All right, good evening. Thank you for your questions. I would like to answer them. So these questions are from chapter 4 through 6 of A World from Dust, and I'm just going to jump in and talk about them. They're not necessarily in order, they're just in the order in which I chose to answer them. What's cool about this is I do have a specific reference that I can point you to for each of these. So this is a question about the human gut microbiome. Maybe this is the story about the guy who was brewing beer in his own gut, things like that. And there is a connection between obesity and microbiome. Now, Dr. Douglas is the one who really knows about these kind of connections, and I'd kind of like to ask him. But I have heard of some things like this question. If you transplant microbiota from a person who is a fast metabolizer, and could some of that fast metabolism be passed on to the other person? Is this a matter of maximizing diversity or of having certain kinds of bacteria? Certain treatments focus on different types of bacteria to one's microbiome. Well, I have a whole article on this, and in fact, I can post this for you. This is from New Scientist. It's about you and your microbiome, and it has several different questions about what's going on with the microbiome, including this little snippet of news on the side. Does the gut microbiome influence body weight? The answer is yes, at least in mice. It talks about one case where there's a scientist who studies this, transplanted gut microbiomes into pairs of identical human twins of pairs of identical human twins. One was obese and the other lean, into germ-free mice, and the animals given the microbes from the obese twins gained more fat mass. In other words, this worked, exactly like what you're saying, but it worked in a human-to-mouse transfer. Notice this is 2013. So people have had time to follow this up, and it's been more complicated to follow up in humans. You can make it happen in germ-free mice. In humans, there is a thing about fecal transplants. I mean, it sounds gross, right? But it really is exactly what you're talking about. Clinical trials have not been able to get this to work in humans yet. There's so much in the humans. Germ-free mice are one thing. They're a clean slate for the microbes to move into. Humans, it's much harder to accommodate the new microbes for them to get a ground. And I guess that's good because bad microbes can't get a foothold as easily. But it does mean that therapies involving microbes have some things to figure out. It does seem like the proof of principle from the mice says that this could work. So I would not be surprised if they found a way to get the microbes actually introduced, just like they found a way to transplant stem cells or to transplant livers or things like that. There's something you have to do to clear the way to get it to go in. Probably something like that here. The mouse study is very suggestive, but it's been very hard to get to work in 10 years. We haven't been able to do it. But great idea, because people are already doing that. This is about an origin of life chapter. It's about chemical gradient inside the rocks, similar to the chemicals, the gradients used for ATP production. So what the thing is, and this is a big part of biochemistry, it talks about the mitochondria work by having a different number of protons inside than outside. They actually pump protons across, and then they let those protons back in, and that movement of protons is what drives ATP production. And that seems like a really complex, it took them forever to figure out even that this thing existed. It was one of the things they didn't really figure it out till the 60s, and it took a really brilliant guy, I think his name was Peter Marshall. He had a common name like that, and so I talked about him in Biochem too. And so, just figuring this out took a long time. So if figuring it out took a long time, how could we even suggest that something like that could have happened with the origin of life? Well, the answer is there are some theories, and there's some surprising similarities, especially when you look at the alkaline vents that are flooding into an acidic ocean, an ocean that would have been more acidic early on, and that forms a chemical flow system that actually looks a lot like the chemiosmotic gradient of protons that produces ATP. And this is what Nick Lane talks about this a lot. I mentioned him a lot in Biochemistry 2 this year. And there are some videos about that, actually. Actually, if you look online for the test, is it test 2 or test 1? It's test 1. Test 1, where I covered another paper of Nick Lane's, that was more about metabolism, but he has a lot of great ideas about this, and I mentioned him, I believe I reference him at this point. This is a figure from his actual paper about it, and I just want you to see those rocks. Those are rocks that are right there. They are sort of porous to protons and to CO2. And you see how you have acid flowing over the surface, and you have alkaline flowing within the rocks. And that could set up something that looks exactly like a cell, where you have OH minus inside the little cell-like compartments, but you have H plus on the outside. And that's the kind of thing that is exactly like the pattern that makes ATP in the cells, in the mitochondria, I should say, because it's inside bacteria and inside the mitochondria. So it's really cool. You can look at the bottom here. There's actually a suggestion of how hydrogen that is produced by the vents is used to produce methyl groups that can be used for like fuel type purposes. There's also actually a production of ADP to ATP, which could happen if the rocks were rich in phosphate, something like that. Phosphates could be put together like this. There's lots of parts of this that are left out. You know, where does the adenyl group come from, the A of the ATP? But this has a surprising number of the pieces present. So even something as complex as the chemiosmotic process, the way that mitochondria work, appears to have some kind of analog in the way the alkaline vents flow. The good news is I know Nick Lane has a lab. He's working on these things. He's actually doing experiments to see what works and how it works. And so I do hope to hear more about this. Even this idea, though, is about 10 years old, is 2007. So, and he has made some other progress since then, but it seems like the experiments are easier going in some other areas. But I still think that this is a great idea, and I hope that it works out. Here's another figure that shows, I know that the person who asked this is on the chemistry side of things. And so this just shows you the acid ocean on the top, the alkaline fluid on the bottom, and the exact change in pH, the difference in pH units. You can calculate out the energies, and the energies appear to point in the right direction. You know, not everything's worked out, but it's not impossible, you know. It's an interesting idea, and I think it's very provocative. And then there was a question asking me to elaborate more on two concepts that were introduced. The first was the ERD, the energy rate density. And I just want to point out that this is Chaison's concept. I introduce it first in this part of the book, and it will keep coming back. So don't worry, I will give you more examples of it as we go along, and then I'd like you to keep this in mind and ask the question again once we get through these later chapters, once you see it again, and it will definitely show up. It even shows up in chapters 11. I think it shows up in chapter 12. Definitely chapter 11 is a big chapter for the ERD. And so the one thing I want to say about it is not a static entity. It's an entity of flow. And it's why it's measured in watts, not in joules. And it's a different kind of thing, but it's the kind of thing that Prigogine was talking about. Then there's another question about one of my favorite discoveries. I had never heard of Winogradsky columns before either. And then I found out about them, and I found out that there's a bunch of people who are really inspired by these because they're really pretty amazing that nature will do something like this sort of on its own, or you can tend it just a little, and it's like a little garden that you tend, but it's a garden of microbes. And they all cooperate, and they self-develop. Also, depending on where you start from, the microbes that you start with can give you very different results. And so there's a whole world here of things to do. And I think there's communities of people who are like Winogradsky column hobbyists. There are scientists who are microbiologists who sell Winogradsky column materials. And then there's the Joyful Microbe, which is a blog I subscribe to. I get news updates from them about every week from her. I forget her name, but you can look up the Joyful Microbe. And in fact, you can find them pretty easily because they even have a shop that sells stickers that looks like this. joyfulmicrobe.com, and it's a little Winogradsky column sticker that you can put on your water bottle or things like that. What I like about this is even this shows the oxygen and sulfur gradient on the left. You see it right there? And it has the different kinds of bacteria that can form. Now, this is a very colorful example. This is sort of an idealized example of what happens. I've tried it myself, and yeah, I got like two colors, and they were both green. It was like green and brown. It wasn't quite like this. I think that you have to be a little lucky, and you also have to try a lot of things to get like the oranges and purples, and even pinks. But Joyful Microbe has little worksheets like this. This is about different ways to make it, and you can keep track of what you try. The important thing is you always have to have a sulfur source. So you have to have like egg yolk, which has a lot of proteins that have sulfur in them, or even some literal sulfur itself. Some people put like a sulfur chemical directly in there, although that might be a little bit too much for some microbes. And you always have to have light. You have to have the flow of light that flows through the system. These are the pictures that you get. The HHMI has a big poster about this. You can look that up with just a quick search. But they have these examples of the Winogradsky columns that you can make. And these are from different people. And you can see all the colors you can make. There's one that I wonder how they got that orange and purple one. The orange is clearly some kind of iron, I think. And the purple, I don't really know. These are the best examples. Mine looked more like the little green and brown ones in the back. But you have to keep trying. And you can, once you find something that works, you might be able to repeat it. Or it might even just be a fluke that you caught a certain microbe at a certain time and it started to grow. That's what I like about it. There's a order in it that develops dynamically, but it's also, there's some unpredictability in it. There's layers of predictability and unpredictability. And that's one of the points of my book, that it's nuanced. And you can't really say it's completely ordered or completely disordered. It's something that's very interestingly between the two of them. And so I just want to share one more thing that happened to me as I was driving into campus. I've mentioned in class Jeremy England. He's the one who's a physical chemist who's actually following up on Prigoghine's ideas. He's also an Orthodox rabbi. And so he talks about, and he wrote a book that talks about his understanding of the Hebrew Scriptures and how they fit with how he understands physical chemistry, which are like two of his favorite things. And I'm like, this is like my favorite guy now, and one of the things that amazed me as I was listening to the interview is he mentioned something that was exactly like this diagram. You remember this diagram from A World from Dust? This was, I want to give credit for this. When you see diagrams like this, these are by Mary Chafee. The Gala Bent does the ones that are more sort of schematic. The ones that are shaded are usually Mary's. And I want to say that Mary actually gave me the originals of these, and so I have them hanging up on my wall. This was actually a figure that was added late in chapter 4. I actually went back and I added this in. I hope it doesn't stand out too much. But I just think these are just wonderful pictures, and I have all 4 of these. And you can see the whole point is you have this, both of these are alluvial patterns. The one on this side, this one is Titan. This one is Mississippi Delta. But the whole point is it's a little bit hard to tell which one's Titan and which one's the Mississippi Delta. You can see the farmland on the Delta. That's one of your clues. But the thing is the branching patterns look the same. And so I had that in my book. I wrote that in 2016. Well, in 2020, there's this Jeremy England was being interviewed, and he was talking about this. You can see right here, he is also an ordained rabbi who finds resonances between modern science and passages in the Hebrew Bible. He talks about entropy fluctuation theorems, how scientists should approach religion. He does name drop Prigogine at one point as one of the progenitors of the field, but he is really advancing Prigogine's work. And I just want to say, he's being interviewed. Sean Carroll wrote a big book called The Big Picture about how his atheism makes sense of the world. And they're in conversation here. And I just think that that's sort of an image that you'll get in my book in chapter 12. The image of the conversation is going to be big in that. But too many spoilers. I just want to read to you exactly what I heard on the way in. Because England is talking about when Moses is given a sign by God of taking the water of the Nile River and pouring it on the drying ground and it becomes blood. Well, that sounds like a parlor trick to a certain kind of ear. But if you take it more seriously, as knowing about what the world is and say, What kind of point could be hiding there if we assume, in a sense, that the text knows how the world works, and it's not dumber than me, but maybe it's smarter than me? It's this kind of attitude, by the way, that can make a text come alive. That when you start to realize is, this text of Moses is a very compact metaphorical discussion of what it is for a non-equilibrium system to explore a space of possible combinations. Doesn't that sound like prigogine? Because the dust of the earth is, yes, a particulate matter that can be stuck together and in different ways, and even dust mixed with water gives you different kinds of mud, different qualitative properties, just depending on the kind of dust and the ratio of dust to water, etc. So you have all these ideas of qualitative emergence and novelty through combination, and it's also perfectly river water, and that the text is talking about not just water. River water is water that flows. So you see, flows are important here to England's thinking, Prigogine's thinking, and to A World from Dust. It's water that carries energy as well as mass flow, and as a result, we even see in the world complex emergent branch structures. If you watch water roll through sediment, you can see the emergence of complex branching that reminds one of the veins of a leaf, or even your own body. And those are the two other images that we have in that. So it's exactly the 3 of the 4 images that we have. It's not exactly the same process, but it's evocative of much of the same physics that we end up wanting to talk about, which is how energy flow can cause particular specialized pages to emerge in the space of combinations that are being explored by a bunch of different particles that can clump together in different ways. England models these on a computer, and he finds that there are ways in which the system can sort of capture the energy flow, and the energy flow shapes the shape of the system, so it's not just a random gas, but the randomness is actually arranged into order. And so that is what I want to leave you on tonight. Thank you for your questions. Have a good night, and good luck studying for the test on Friday.